Well, hello everyone, and welcome to this very first combined webinar between two great societies. So this has been organised by the International Society for Stem Cell Research and the World Parkinson's Coalition. And this is the first time that we have done this. So you're very welcome, and we're very much looking forward to your company during this. This has been co-sponsored by Blue Rock Therapeutics. We're thrilled to have you join us for this open access programme. My name is Roger Barker, and I am a professor at the University of Cambridge in the UK. I'm the president of the World Parkinson's Coalition and a member of the ISSCR Education Committee, and I was a previous board member of the ISSCR. I will be the moderator for today's programme alongside Eli Pollard, who's the Executive Director of the WPC. Today, I'm joined by two fabulous speakers, Agneta Kirkaby and Vivian Tabar, who will present on the latest research and clinical advances in cell therapies for Parkinson's. I'm also delighted that we've got Michael Reinberg here, who will share his personal experience from receiving a cell therapy for his Parkinson's disease. This is a wonderful, great opportunity to engage with both scientists and clinicians, as well as a patient with first-hand experience. We encourage you now and throughout the next hour and a half to ask questions throughout the program. Please submit your questions in the Q&A tab at the top right hand of your screen at any time during the presentations, and we'll try and get to as many questions as we can following each presentation. So you're very welcome, and it's now my great pleasure to pass the floor to Keith Arm, who's the CEO of ISSCR, and Eli Pollard, who's the Executive Director of WPC, to share a few words.
Roger, thanks very much. On behalf of the International Society for Stem Cell Research, I just want to send a big welcome to all of you. Thank you for being here. You know, for more than 2 decades, the ICCR has promoted excellence in stem cell research and its applications to human health. And that's exactly what we're here to do today, to gather researchers and clinicians and patients and their families to help advance the field forward, especially as it relates to Parkinson's disease. And so we are so tremendously proud to partner with the World Parkinson's Foundation on this important program. The World Parkinson's Foundation is such a tremendous advocate both for the research but also for patients and their families. So we feel very fortunate to be able to collaborate between organizations like this. To our speakers, thanks so much for being here. We can't do this without you, of course, and to all of you, thank you for joining us. It is gatherings like this that give me a great deal of hope for the future. Eli.
Thanks, Keith. Hi, everyone. I am so pleased to co-host this webinar with the International Society for Stem Cell Research. The WPC, for those of you who know us, you know we've been working in this space for more than 20 years. And those of you who are meeting us for the first time, for us, it's so incredibly important to find ways to bring the community of researchers, clinicians, and people with Parkinson's together so that we can both learn with each other and from each other. And we've been doing that for more than 20 years. So this opportunity today is just outstanding. We will know that when people get out of their silos, their research silos, their clinical silos, it allows for unique relationships to grow and for innovation to bubble up. So the topic of stem cells and Parkinson's is the perfect place to bring our communities together to advance our collective understanding of this area of research. And for those of you who are joining, who are living with Parkinson's and joining us today, and those of you who are just non-researchers, a big hello and welcome to you, especially if this is the first scientific webinar you've joined on this topic. You are in the right place today with exceptional presenters lined up for you, where you're going to hear, you know, the cutting edge research around cell therapy. And thank you for joining us. I'm handing back over to Roger.
Well, thanks very much, Eli. Thank you very much, Keith. And thank you all again all very much for joining us today. I would like to start by thanking the co-sponsor of the webinar, which is Blue Rock Therapeutics, and I'd like to share their sponsor message. Blue Rock Therapeutics LP, a wholly owned and independently operated subsidiary of Bayer AG, is a clinical stage cell therapy company focused on creating cellular medicines to treat devastating diseases. BlueRock is harnessing the power of cell therapy to create a pipeline of new medicines for people suffering from neurological, ophthalmic, cardiovascular, and immunological disease. The content of this webinar has been developed, however, by the speakers and has not been influenced by the co-sponsor of the webinar. So now let's begin the webinar, and I will start by setting the scene by giving a brief overview of dopamine cell therapies for Parkinson's, why they're being considered, and just a little bit about the background to this topic. So I'm now just sharing my screen, and I hope that that is now in presentation mode. It's completely vanished off my screen. I'll try. That's in presentation mode. I hope you can all see that in presentation mode. If there is a problem, just give me a shout. So what I want to do is I just want to introduce you to Parkinson's, and then a little bit about the rationale for why we're using dopamine cell therapies. So Parkinson's disease takes its name from James Parkinson, who described it in 1817. And it is characterized even in 2024 by the clinical features of the disease. We have no diagnostic test for it, but there are a classical triad of problems which people present with, which is arresting so-called pill rolling tremor, slowness of movement, stiffness in the muscles, typically with postural instability, i.e. slight difficulty keeping one's balance, and a host of non-motor problems. So it's a very complex disease which has at its core the loss of a specific population of cells within the stem of the brain in a place called the substantia nigra, where on each side of the human brain we have between 400 and 500,000 dopamine cells. And when you've lost half of those, namely 200,000, you start to develop the early features of Parkinson's. That was discovered well over 50 years ago and led in the 1960s to the first dopamine therapies for Parkinson's, which remains the mainstay of therapy in 2024. So essentially, as people are diagnosed and then progress through the condition, a whole host of drugs are used, all of which target the dopamine system. So there are dopamine agonists that target the dopamine receptors. There are inhibitors of the dopamine breakdown, monoamine oxidase inhibitors, catechol-L-methyl transferase inhibitors. There's L-DOPA itself, which replaces the dopamine. And then there are infusions of dopamine, i.e. apomorphine or duodopa or produodopa, and similar drugs. There are surgical procedures which are also used, but essentially the surgical procedures and the amantadine are all used to treat some of the complications which arise from the dopamine drugs, which in the early stages of the condition are an extremely effective therapy, such that patients can almost look as though they've gone entirely back to normal. So that's important for our discussion today, because it shows that there's a clear therapeutic target, the dopamine system. So the question arises, why use drugs? Why not just replace the 200,000 dopamine cells which are lost as part of the disease process? So then the question arises, well, if that is the case, what dopamine cells could I use to replace those? Now, clearly one could try and repair the system from within. Can you turn on new dopamine cells within the brain to replace those that are lost? Clearly that doesn't work, otherwise you wouldn't have Parkinson's. And whilst there are a number of ways in which people are approaching this, essentially none of them have yet really progressed to the point where we're thinking about how we can do this on a bigger scale. There have been attempts to turn cells into dopamine factories using gene therapies, but for our discussion today and where the field is currently largely concentrating is around the concept of replacing the lost dopamine population by putting in a new group of dopamine cells. So why would you want to do that? So why would you want to put new dopamine cells in when I've already told you there are effective dopamine treatments which are in the clinic and have been in the clinic for over 50 years? Well, I think there are three major reasons why one would consider this to be a good way forward for treating this condition. The first is that by putting the dopamine cells into the brain at the site where they're lost, or where the dopamine major dopamine loss occurs, you will avoid all of the side effects, or many of the side effects that you see with the drugs that we use in the clinic. These include stimulating bits of the brain which have normal dopamine levels, so they give people neuropsychiatric problems, they can hallucinate, they can develop abnormal behaviors, they can start gambling and such like. by using this, you would avoid all of the side effects that you would see with the current medications, which don't occur in all people, but happen in a significant number. The second major advantage would be that if you put back dopamine nerve cells at the site where they are lost or the place where they work, they can release dopamine in a normal physiological way as they would in the normal brain. And that would avoid one of the long-term complications you see in patients who take L-Dopa, which in the UK is drugs like Sinemet and Madapar. And that is that after a number of years, the drugs themselves produce side effects in terms of involuntary movement, so-called dyskinesias. And in the previous slide where I was talking about amantadine and deep brain stimulation and focused ultrasound treatment to make little holes in the brain at strategic sites, all of those are primarily designed to treat this complication of the L-Dopa medication, namely L-Dopa-induced dyskinesias. So this treatment would avoid those side effects, and as such, it would avoid the need for those therapies. And finally, of course, one of the great attractions of this is it would actually be a one-off treatment. You would have the cells replaced with this transplant. You wouldn't need anything else. And whilst it would obviate the need for taking any dopamine drugs, of course, it's not, it's only repairing that bit of the brain, but it would avoid all the complications you see with those medications. So this therapy has the potential to transform what we could do in Parkinson's. There are a few things it's worth just mentioning, just so that people understand the limitations of this treatment. Obviously, in Parkinson's, there is pathology which extends outside the dopamine system, the nigrostriatal dopamine system. There are a whole host of other symptoms and signs that patients have which reflect pathology outside of that system. This treatment will not touch those. It may reduce the size and strength of these symptoms by reducing the side effects which some of the medications produce in these areas. But fundamentally, this is only treating one, but a major aspect of Parkinson's disease. Secondly, it's not going to suit everyone with Parkinson's because really this therapy will work best in people who have a very good response to dopamine. Sadly, some people don't. Some people have major problems outside the dopamine system. This therapy would not be suitable for them. And perhaps importantly, it's to understand that this is not curing Parkinson's because simply all one's simply doing is actually treating one aspect of it. The disease pathology, the accumulation of the protein in the brain, the alpha-synuclein will continue. So it's not a cure, but it would be a transformative therapy. So the next question then is, if we're going to take this approach, what type of cells could we use? Well, a large number of dopamine cells have been considered. There are dopamine cells which can come from other sources in the body, so things like the adrenal gland, which sits near the kidney, not very effective. There's been a lot of interest in using the dopamine cells that develop out of the human fetus, so using fetal dopamine cells, which has been the source of the most successful transplants over the last 35 years. What we'll be concentrating on today are the stem cells, so both embryonic stem cells, which come from spare embryos in IVF programs, or reprogrammed adult cells, so-called induced pluripotent stem cells. This has not been a field that's been idle for many years. It's been a very active field for almost 40, 50 years now. And in the early days, a lot of the work was concentrated on using cells other than the developing dopamine cells. But actually, from the 1980s right up to the last decade, human fetal dopamine cells were used to try and repair the brain. But of late, and where our talk will be going today, is on the stem cell therapies, which are now emerging out of the labs and in the clinic in a very exciting time for Parkinson's disease. So you'll be hearing in this webinar today, we have Agneta Kirkeby, who'll be telling you how we can take a stem cell and turn it into a dopamine cell for use in a clinical trial. We've then got Michael, who'll be talking about what it's like to be the recipient of a dopamine cell therapy for his Parkinson's. And then we will conclude by Vivian telling us where we are with clinical trials with stem cell dopamine cells and where that field will be going in the future. So that's just by way of trying to introduce the topic so you understand how we've got to today. I'm now very happy to take any questions that you might have for me.
Roger, I do have one question for you. Great talk. Thank you for that introduction. So, we have a lot of great, dopamine therapies already accessible to people with Parkinson's. So what are your thoughts? Do you really think we need a dopamine cell therapy with what we already have on the market?
Yeah, I mean, it's a good question because it's a common question. We have very good dopamine therapies. Why not concentrate on something else rather than dopamine? I think the answer is that dopamine clearly is still the best therapy for Parkinson's. Since it was introduced in the 1960s, it is still the most effective treatment we have is L-DOPA. If we could simply put L-DOPA back in a better form, and that's happened with some of these infusion therapies over the last 10 to 20 years, clearly we get better control of patients with Parkinson's using that, but they still have to have pumps, they still have to have cannulae, they still have to titrate it. This would give you the opportunity to put in a cell. It's a one-off repair. It fixes that network. And in theory, if it worked, then everything we give for Parkinson's in 2024 for the motor problems would essentially not be needed. So it could be transformative. And I think if it works and it's affordable, which is obviously a question that a lot of people ask me if it ultimately works, then I can see it being a therapy which would transform what we could do in Parkinson's.
Yeah, I have a couple other questions for you, and that was a great response. Could you go back? You said in the talk earlier that this is not going to cure Parkinson's, but what it is going to do. We have one question in the queue about why doesn't this actually cure Parkinson's? Can you just explain that again?
Yeah, so basically to cure a disease, you have to get rid of the problem. So if you have a pneumonia, you are cured of it because you take an antibiotic, it kills the bug that's causing the pneumonia, and the pneumonia is gone. So that is the end of it. In Parkinson's, the fundamental problem, which is unclear why it happens, is there's a protein in your brain, a normal protein, which becomes abnormal and aggregates, and that's called alpha-synuclein. So that pathology will carry on, and that will be carrying on despite the fact we put the new cells in. So we're not curing the disease. It is, however, important. So if you cured the disease, obviously that would be the end of it. But one of the things people often talk about is disease modifying. And disease modifying often means targeting what causes the disease and stopping it. It's also very important to realize that good symptomatic therapy has a major effect on prognosis. So if you were in the 1950s and you had Parkinson's, your life expectancy was probably no more than 10 or 15 years. Nowadays, your life expectancy at the community level is normal with Parkinson's, and that's due to the drugs that you take. Most people with asthma are never cured of asthma, but they have very effective inhalers, which essentially means they live a normal life with their asthma. So good symptomatic therapies can have disease modifying effects, but fundamentally the stem cells are not removing the pathology that underlies this condition.
Thank you, Roger. We have quite a few questions, actually. I'm just going to finish on one to keep us on time, is from our friend Jagdeep in the UK. Is just asking about STEM PD trial. Any sense of a timeline, Roger, when that will be complete with results available?
So the STEM PD trial, and I don't want to take anything away from Agneta, who might be talking about this. This is a European trial which involves 8 patients and is a trial conducted between Lund in Sweden and us in Cambridge. 8 patients, I can say, have now completed the transplant arm of the trial. The data will probably not be available for at least a year and a half, because we have to get all patients to a certain point before we can report on it. We're hoping to put out a paper which outlines how the trial is designed, but actually the results from it won't be available for some time.
Thank you, Roger. I think we're going to conclude with your Q&A session and move on to Agnete. Thank you so much.
Thank you, Eli. And now it is my great pleasure to introduce a great friend of mine and a wonderful colleague who's been working on this field for many years, Agneta Kirkeby, who's from the Novo Nordisk Foundation Centre for Stem Cell Research. I should say that's an independent centre for research. That is not Novo Nordisk have given the money for it, but it's not Novo Nordisk itself. She's at Renew in the University of Copenhagen, and Agneta is going to tell us, and has not only tell us, but has done this herself, how we turn a stem cell into a dopamine cell for use in Parkinson's disease. So thank you very much, Agneta, for joining us today and telling us about this.
Great. Thank you so much, Roger, for that introduction. Yes, so I'm going to go a little bit into some of the nitty-gritty details about how we do this in the lab. And this is really some work that started many years ago, around 2009. I'm just going to share my screen here. Yeah. Okay, so I hope you see everything okay. So as Roger mentioned, I'm at University of Copenhagen, but I'm also at Lund University in Sweden, which is actually where this work has been done and where the clinical trial, the STEM PD trial, is currently ongoing. Just a few disclosure statements. I am a co-inventor of several patents related to dopamine cell products, and I'm a consultant for several companies. And we partner with Novo Nordisk on the development of the STEMPD product for use in Parkinson's disease. So, as Roger already introduced to you, one main hallmark of Parkinson's disease is the loss of dopamine neurons, particularly in the substantia nigra. And these neurons are critical for conveying signals coming from our motor cortex and conveying these into actual function of our limbs. So when these dopamine neurons are lost, this communication between our motor cortex and the rest of our body is not functioning properly. And this causes this rigidity, slowness of movement, and tremor, which is cardinal symptoms of Parkinson's disease. And as the disease progresses, more and more of these dopamine neurons in the substantia nigra, and this is what you're seeing here. This is a brain section showing you the substantia nigra. It is called the substantia nigra because it is black. These neurons are contain a black pigment, so you can see them by eye. And in a Parkinson's disease brain, this pigment is completely lost because the neurons are lost. So put quite simply, the idea of cell replacement therapy is just to transplant new dopamine neurons from the exterior into the brain to replace the function of those dopamine neurons that have been lost. Now, this is actually some work that started many years ago, even before we had access to the stem cell technology. So already back in the 80s, trials of transplanting or studies of transplanting dopamine neurons to the brains of Parkinson's disease patients were performed at Lund University Hospital in Sweden. And what they did here was they took small subdissected pieces of fetal brain, that particular piece of the brain that they knew would give rise to the dopamine neurons. This was really pioneered by two very well-known scientists at Lund University, Anders Björklund, Olle Lindvall, and also with Patrick Brundin. And they took this very small piece of tissue from the fetal brain, and this was tissue that came from legally donated tissue from abortions. And this is the piece of the brain that we know gives rise to dopamine neurons. So you can think of this as kind of a tissue donation. You take this tissue out of the aborted fetuses, and then they could make it into a cell suspension and transplant it to the brains of Parkinson's disease patients. And they transplanted the first patient in Sweden in 1987. So this was really a way to test this concept before we could produce stem cells in the lab, to test whether it's actually possible to transplant new dopamine neurons into the brains and whether they can survive and whether they can function. So these were very important studies, and they've laid the groundwork for everything that we're doing today. I just want to point out here, and Roger also mentioned this, the cells are transplanted into the striatum or the putamen, which is the target site of the dopamine neurons. They're not transplanted into the substantia nigra, and that's because this is where we want them to function, so they're closer to their target. So what we saw from these transplants was that there was a lot of variation, but some of these patients really had very remarkable effects of the transplant. So this is an example of two patients from the Lund cohort, patient 7 and patient 15, which received two grafts, one of each side of the brain. And they really had clinical effect of these dopamine grafts for many years and could even go off dopaminergic medication for more than 10 years after transplantation. So we learned here that when these grafts are working, when there's enough cells and they're transplanted correctly, these transplants can really have good beneficial effects, at least in some patients. What we also learned from these trials was that the graft could survive in the brains of the patients for many years. This is an example of a 24-year-old graft. This is the oldest graft we have available in Lund, showing a healthy looking graft in the putamen of a transplanted patient now 24 years after transplantation. And this patient was off immunosuppression for more than 20 years. So this shows us that the graft can survive for many years even without immunosuppression. And what could also be seen in these patients was that on fluorodopa PET scanning, which is a commonly used PET scan to look at dopamine function in the brains of patients, you could see a signal from the graft post-transplantation. But what you could also see was that this was seen often in these patients, that one graft was typically better than the other. Typically, there was never enough cells to transplant both hemispheres on these patients, so they would receive one graft on one side, then wait some time, receive another graft when there was enough tissue, and often there was variability in the graft survival and graft function. So this really illustrates the challenges with this approach. It was very difficult to standardize the quality of the tissue, the amount of tissue in these transplants, but when there was enough tissue, there was a very significant effect in some patients. So if you want to learn more about this, you can read this excellent review written by Roger, summing up the results from various clinical trials taking place not just in Lund, but also in the US and other places in the world. And in short, what we learned from these trials was that transplanted dopamine neurons can survive for more than 20 years in the Parkinsonian brain. The effect of the dopamine neuron replacement is slow, but can give substantial long-term clinical benefit when it's successful. But that this fetal cell donation, the quality is very variable, and in addition has ethical and logistic hurdles, so you can't scale it up to many patients. So this, of course, led to the quest of looking for another source of dopamine neurons, which would not come from donated fetal tissue, but instead from some kind of stem cell where you can do unlimited production in the lab. Now, we do actually have stem cells in our bodies. We do have stem cells in our skin, our blood, our muscle, our intestine, but these have very particular function. Skin stem cells can make skin if we get a wound. Our blood stem cells can make blood. Our muscle stem cells can make muscle, and so on. They can't suddenly go on and make another tissue than the tissue they're intended to make. And if we look at the brain, unfortunately, the situation is that we have very few stem cells in our adult brains, and those few stem cells we have cannot replace our dopamine neurons. So what do we do in these cases where we don't have stem cells in our adult bodies? Well, we go and search for a stem cell which can make dopamine neurons. And it turns out to find such a stem cell, we actually have to rewind time all the way back to the very, very first days of our human development, around 4 days after egg fertilization. We have a structure called the blastocyst. So the blastocyst is a ball of cells that you get 4 days after the egg has been fertilized. And these are, for instance, produced at IVF clinics as a normal part of the IVF procedure. Now, this is what a blastocyst looked like. And in the middle of a blastocyst, you have what's called the embryonic stem cell, also called the pluripotent stem cells. This is the size of a blastocyst at this stage. So you can actually take these cells out of the blastocyst, and then you can culture them in the lab. And this was a technology that was discovered in 1998. And here it was discovered how you could culture these cells in the lab and keep them proliferating forever and ever. And then you get a culture that looks like this. These are embryonic stem cells, looked as what they look like under the microscope. They're like, make colonies like this. And each cell is like tiny, tiny cell that is embedded within these colonies. We also call these pluripotent stem cells. Pluripotent means that they can make every single tissue of our body. And that is because they come from this very particular snapshot in development where these cells are intended to make every single tissue of our body. So pluripotent stem cells are quite amazing, not only because they can proliferate indefinitely, but they can also make every single tissue of our body, and including the nerve cells and the dopamine nerve cells that we're looking for to replace the dead cells in Parkinson's disease. Okay, so with a human pluripotent stem cell, you can produce a midbrain dopamine neuron in theory and use it for transplantation. And you can get a human pluripotent stem cell either from a blastocyst or through a newer technology that we call reprogramming that was invented in 2007, and for which the responsible scientist won the Nobel Prize. Here you can generate a stem cell from any living individual through taking a fibroblast or a blood cell, a blood sample, and then you can actually make a human pluripotent stem cell from that. So whichever route you take, when you have a pluripotent stem cell, you can make it into a dopamine neuron. But how do you do that? Well, here's one challenge. One challenge is that the human brain contains many different subtypes of neurons, not just dopamine neurons, and there are actually more than 1,000 different subtypes of neurons in the human brain. So this is one thing that we and many other stem cell scientists have been struggling with for years, is figuring out how you go from a human pluripotent stem cell into one specific type of neuron. And in this case, we're interested in making the dopamine neuron. So what we do in the lab is we take these pluripotent stem cells and then we add different combinations of growth factors in order to get the cells that we're interested in. And to make a dopamine neuron, we need to make cells coming from the ventral midbrain, because the ventral midbrain is that part of the embryonic brain that will form the dopamine neurons. And remember that this is also the part of the brain I told you before, which was transplanted into those patients in the 80s in Lund in Sweden. So if we can make this part of the brain, then we can also make the dopamine neurons. So basically, what stem cell scientists spend a lot of time doing in the lab is just adding different combinations of growth factors at different concentrations at different time points and seeing if they get the right cells. In this case, several different groups have been working on this across the world, and the consensus is that it requires around 7 to 10 different factors to make ventral midbrain progenitors, and you can check the quality of them by looking for certain markers, in this case, 2 markers called FOXA2 and LMX1A. And if you do it right and use the right combination, you can get very high purity of these ventral midbrain progenitor cells. You can take these progenitor cells and mature them in the lab by adding maturation factors, and then you get nicely looking dopamine neurons that also show action potentials like an adult functioning dopamine neuron would. But when we're talking about transplantation therapy for Parkinson's disease, we're actually not so interested in looking at how the neurons function in the lab. We want to see how they function if we transplant them. And here it's important to note that what we transplant is not the mature neurons. What we transplant are the progenitor cells, which are kind of an immature cell state where they have decided to become a dopamine neuron, but they're not yet a dopamine neuron. And the reason for that is that you can't transplant mature dopamine neurons, because then you start breaking up these long processes that the cells are sending out. And this is really detrimental to the cells when you transplant them. So we transplant the immature progenitor cells, and then we look at how they mature in vivo and whether they start forming dopamine neurons. And the way we test this is we take a rat model where we inject a toxin on one side of the brain to kill all the dopamine neurons on one side of the brain. So we have a rat with an intact dopamine system on one side and a completely lesioned dopamine system on the other side. And that's what you see here. This rat brain has been stained for dopaminergic terminals. And then we transplant our stem cell-derived progenitors into the brain. And then some 6 months after transplantation, it looks like this. If you use the correct differentiation protocol to get your ventral midbrain dopamine progenitor cells, you can get a brain that looks like this, where you have a complete re-innervation of the dopaminergic nerve terminals. In contrast, if you leave out one of those 8 to 10 factors that you use for differentiation, you get another type of neurons. In this case, we made cortical progenitors. Here you get a graft that's full of neurons, but none of these neurons are dopaminergic. So this graft would be non-functional. Now, to test whether these cells are functional, this just to mention, we tested this in more than 3,000 rats, and these grafts, even though they are proliferative when we transplant them, have a very good safety profile. So we've never seen a tumor or any other adverse graft events in these animals. So what we need to test, of course, is whether they can release dopamine, because this is, of course, the main job of a dopamine neuron. to release the dopamine. This is the main cause of Parkinson's disease symptoms, rigidity and lack of movements. It's because the degenerated axons and neurons are no longer releasing enough dopamine. So to test that, we use this rat model, which has been lesioned on one side for dopamine neurons. And then we give these rats amphetamine. And what happens when you give it amphetamine is that you have this massive release of dopamine from the intact side. And because you now have an imbalance in dopamine release, what happens is that the rat starts rotating around itself. So it has a lot of dopamine release on one side and no dopamine release on the other side, and that creates motoric asymmetry. And then you simply count how many times the rat rotates around itself after you've injected amphetamine. And then we transplant to the lesion side. And what we see then over time, and it's important to say here, this does take actually several months before the graft becomes functional. Over time, the rats that have received a graft with the dopamine stem cells will stop rotating, and in some cases even start rotating the other direction, depending on how many stem cells you transplant. And this is really a measure telling you that the dopamine neurons you've transplanted are functional and are releasing dopamine and are communicating with the rat host neurons. So this assay tells us that the dopamine neurons are working and they're releasing dopamine. We've also tested it in a pig model. And the advantage of this pig model is that we can put the pig into a PET scanner, which is the same type of PET scanner that is used for humans. And in this pig model, what we did was we did a PET scan for fluorodopa, which measures dopamine terminals, as well as P2I, which measures the dopamine transporter. And here we could see on the transplanted side at 7 months post-transplantation, there was a clear signal from the transplanted dopamine neurons, which of course is an indication that we can see the graft and hopefully use this assay also in patients. Okay, so that's one job of a dopamine neuron, but another important job of a dopamine is to reuptake dopamine. This is important because dopamine neurons are actually the only neurons in the brain that express the dopamine transporter. So they're the only neurons that can clean up after themselves and reuptake the extracellular dopamine. This is particularly important for patients because many patients are on L-DOPA. And what L-DOPA does is it actually hijacks non-dopaminergic neurons to produce more dopamine. This is good. They get more dopamine in the extracellular space. But the problem is that because the normal dopaminergic terminals are lacking, there's not enough reuptake of that extracellular dopamine, which sometimes causes an overload of dopamine, which is what Roger also talked about. This can cause involuntary movements or L-DOPA-induced dyskinesias. So we tested if the transplanted dopamine neurons could also uptake dopamine. And we did that in collaboration with MLA at Cardiff. They have a model where they first lesion the dopamine system and then give the rats L-DOPA every single day to kind of mimic the clinical situation for the patients. And then these rats start developing L-DOPA-induced dyskinesias. We then transplanted these rats with the dopamine progenitor cells and found that over several weeks, these abnormal involuntary movements completely disappeared. So what this assay told us was that the transplanted neurons not only can produce dopamine, but can also reuptake dopamine in the host brain. So these are really the assays that we've done to test the functionality of the dopamine neurons before going into clinical trial. So this is the overview of many years of work, is basically developing the protocol, the different factors needed for dopamine neuron progenitor production. Then in our case, and also in the case of Vivian, who will be speaking to you later, we freeze down the product, and this is with the purpose of being able to distribute it to several clinical sites. In our case, the cells are cryopreserved and stored in London at a facility there. We do quality control testing of the product, and then we do a range of other studies. We stability test the product. We've done safety study in 130 nude rats. These are rats without an immune system, so they can tolerate the graft. We do an efficacy study to test that the functionality of the clinical product is as expected using the rotation assay I just showed you before. And then we have put together all this documentation into an application for an clinical trial testing 2 different doses in a total of 8 patients. For this MPD product, we transplanted the first patient in February 2023, administering 7 million dopamine progenitor cells on each side of the brain. Sorry, in total, so 3.5 million progenitor cells on each side. We've now completed all 8 patients with 2 different doses, the second dose being double of the first dose. And as Roger said, we're now waiting to complete or collect data from these 8 patients. So the primary outcome is at 12 months post-transplantation, and the secondary outcome is at 36 months post-transplantation. So we're not the only ones, of course. You'll hear from Vivian in a bit. Here's just an overview of the currently ongoing dopamine cell replacement clinical trials. So we have our trial, which is ongoing in Sweden and the UK. We have the Blue Rock trial, which is in New York and Canada, and which was just completed. Then we have a trial in China, a trial in Japan, which was also just completed, but where the data hasn't been released. A trial in South Korea, which is ongoing, where they transplanted 12 patients. And then we have a single case at Harvard University in the US, where they transplanted 1 patient with autologous cells, so cells that came from the individual patient. This is only possible through reprogramming technology. This trial will be followed up with further patients it was just approved by the FDA, but hasn't started yet. And then there's a company called Aspen Neuroscience that also started a trial with autologous transplantation. So we're actually seeing a lot of development in this field, and we're actually very excited to see the data coming out. And luckily, you'll hear from Vivian soon about the data from the Blue Rock trial. So with that, I just want to thank and acknowledge all the people involved. This has, of course, been a huge team effort with many different people, a collaboration between University of Cambridge, Lund University, and partnering also with Novo Nordisk, who helped us go into clinical trial, and with a lot of funding from the EU. The project is led by Malin Parmer in Lund, and with Roger Barker as our clinical lead, and then many people involved in the preclinical and clinical development. Thank you so much for listening.
Thank you very much, Agneta. Wonderful talk, taking us through the field and how we've arrived at where we have to today with stem cell therapies. We're a bit short of time, but there were just a few questions which came up in the question and answer, and obviously do reply to the others after this. One of the questions is about what you transplant and the sort of homogeneous population. Do you sort for dopamine cells? What do you worry about the other cells that you've put in, and would it make sense to have a process where you could just select the dopamine cell progenitors?
Yeah, we don't sort because basically the product seems safe and efficacious even without sorting. You do lose a lot of cells if you sort, so that is very much a bottleneck. But in the Japanese trial, they are sorting. It is a progenitor population, so upon transplantation, it does give rise to other cells and dopamine neurons. But we haven't seen any safety concerns with this. And many of the non-dopaminergic progenitors we see in the product are actually also cells that we see in the fetal tissue transplants. So we're not so concerned from a safety point of view, but there are different approaches actually to investigate whether you can get a better product through sorting or other strategies.
And sort of related to that, there were a couple of questions around controlling proliferation once they've been transplanted. And obviously, if I've understood it correctly, the protocol is such that the cells are committed to becoming dopamine cells when you graft them. But obviously with the preclinical work, have there been any concerns that there are cells there that are proliferating that shouldn't be proliferating? Or how do you control the proliferation once you've transplanted the cells?
So this is, of course, something we monitor and have monitored very closely in our safety study. And this is something the authorities, of course, have a lot of focus on. The graft seemed to, after 3 to 4 months, seems to stop proliferating. and stabilizes in size. And what you're seeing after that is really just the outgrowth of neurites. And we've transplanted many rats, and we've never seen like an overgrowth or tumor forming in these animals. So even though they are proliferating, it does seem to be a process that just naturally stops some months after transplantation. And we do get much better integration of the cells when we transplant these immature proliferating progenitors than if we were to transplant a more mature product. So that's the balance we're aiming at here.
And just the sort of final question is that there's been an interest in allogeneic, i.e. taking it from one human to another, or autologous, i.e. as you were saying, taking it from the same person, i.e. the patient donating their own cells. Do you see an advantage or disadvantage in each of those approaches, and what would be your sort of perceived best way forward, if you like?
So of course, the advantage of going autologous is that you don't need immunosuppression. The disadvantage is that this will likely be a very, very expensive product because it is just extremely expensive to produce a new product for every single patient. And we don't know yet, but it might be prohibitively expensive when reaching the market. So right now we're seeing both approaches taken. It should be said that you actually don't need lifelong immunosuppression when transplanting to the brain. So in our case, we're doing just 1 year immunosuppression. So we think that's acceptable, but of course, it is still immunosuppression for 1 year. So that's the trade-off here.
Yeah. Well, thank you very much, Agneta. A wonderful talk as ever from you. Very informative. So thank you. And now I would like to introduce Michael Reinberg from Sweden. Michael is going to join me for a conversation about his experience of living with Parkinson's and his receiving a dopamine cell therapy. So Michael, thank you very much for joining us. Obviously, this is not easy because one gets a bit anxious when doing this. I've done this for many years and I'm still anxious, but I thought it would be useful just to begin with talking a bit about yourself, Michael, if you don't mind, how old you are, and before you developed Parkinson's, just a little bit about your life, what you were doing and such like.
I'm 56 years old. I used to play football, a lot of sports before, but no more.
Okay. And what was your job, Michael? What were you doing?
Ventilation technician.
Ventilation. So these were installing ventilation units and such like, or designing them, or yeah.
No, go ahead.
Yeah. So you were enjoying your sport. You were playing football. You were doing this job, and then when did you first notice a problem?
2005, I noticed that I was trembling when I was carrying a cup of coffee.
So how old were you then, Michael? So you must have been about 40?
Yeah.
And when you say fumbling, what was the problem? You were shaking, or you just had difficulty coordinating?
I couldn't, no problem with coordination, but I couldn't carry a cup of coffee with my left hand.
Right. Because it was shaking so much that the coffee was coming out.
Yeah.
And what did you think was the problem at that stage? Did you think there was a problem, or did you think it was just one of those things?
I thought it was from work. What do you call it? You drill a lot. I don't know what you call it, but I thought it was an injury from work.
Okay. So you sort of injured yourself and the hand was a bit weak, and that's whilst it was shaking. So
yeah.
And so what happened? Presumably it didn't get better. And what made you go to the doctor about it?
My colleague at the time, he had read Michael J. Fox's book.
Okay.
So he said to me, you better go and check it out.
Okay. So this was a work colleague?
Yeah.
And then when you went
my boss.
Oh, your boss. Oh, okay. So you got to do what he says, definitely.
Yeah.
So you went off to see the doctor, and what did the doctor say?
The first doctor did say it was nothing. It was normal. But the second opinion was he sent me to Lund, to Håkan.
Okay. So you went then to Lund University, where we were hearing about this pioneering work on the cell therapy, and Håkan Vidner, who's one of the neurologists there.
Yeah.
And so at that point, you didn't think you had Parkinson's. You just sort of thought, I better go along because they're not quite sure.
No, somewhere in my back of my head, I thought about it, but I was hoping that it wasn't Parkinson,
Okay.
but it was.
And so you saw Håkan, and he then told you the diagnosis. And
No,
how did you?
yeah, after a few X-ray sessions and all that thing they went through to check out to find out if it was Parkinson or not.
So you had some scans and such, like a scan looking at dopamine, a DAT scan,
Yeah,
and such.
4
like.
or 5.
Yeah. And so you were then told you had Parkinson's, and what was it like? So this was, you know, in your early 40s, you'd suddenly been told this. I mean, what was your response to that? How did you react to it?
When I got the message, I called my wife and cried.
Right.
So it was not funny.
No.
But
And
you have to live with.
yeah. And so it obviously came as a great shock because you suddenly had this diagnosis, but you were then put on medication.
Yeah. L-Dopa.
Yeah. And how did that do? What was that like for you? Was that good or bad or didn't do much?
No, it did good. Helped me.
Yeah.
I could keep on working for full day, 8 hours a day.
Yeah.
I could play football.
Yeah.
I could do all kinds of sports too. When I got the diagnosis, I could
So did the
play.
treatment would the treatment have put you back to normal? Did you feel you could pretty much do everything you did before you started the medication?
Yeah, almost.
Okay. And then so this was whatever it was, what you said 17 years ago. And then what happened with the medication over the years? Did it stop working, or did it produce problems, or what were the issues with the medication?
Increase the doses.
Yeah.
So it kept the Parkinson in place. So I didn't get worse, but it didn't get better either.
Okay.
I could keep on doing what I was doing.
Okay. And did you find that after a number of years the medication didn't work so well, or was less reliable, or?
No, no, not really, because Håkan is very good. We tried a lot of different medicine, and yeah,
Yeah.
I
So
was a guinea pig.
it kept you pretty good. So when I was giving my talk, and also Agneta mentioned it, after a while these tablets can produce these so-called dyskinesias, involuntary movements. And so did you start to develop those?
One couple of years ago, I had a gambling problem, so we had to change the medicine.
Okay, so that was one of the side effects I was mentioning. So one of the drugs, the dopamine agonist, that caused you problems with gambling.
Yeah.
And then you had to go on to the L-Dopa, and did that. So obviously at the moment you have these slightly involuntary movements, these dyskinesias, and I imagine this is what happened with the L-Dopa as well.
Yeah.
Yeah. So although the drugs were very effective, they weren't without side effects. One of them caused gambling, one of them caused the involuntary movements. And
yeah.
And so how did you get to hear about TransEuro, which is this fetal transplant program?
I was asked from Gesine, Paul. She asked me if I wanted to be a part of it. So first I was a test person, and then they chose me and a couple of other people. So I was asked from my doctor if I wanted to join.
So they asked you, so these are the other neurologists in London, they said, would you like to be a part of TransEuro, which is this fetal dopamine transplant trial to replace the dopamine cells, which we've been hearing about? And what was your response to that?
Positive.
Yeah.
But when I read I had to sign a paper before the operation, and that was thick as the Bible.
Yeah. So when you went into the operation, how were you? Did you feel very positive about this as a move towards the Parkinson's disease, or were you very apprehensive, worried about it? What was your sort of state of mind as you went in to have this transplant?
I was positive.
Positive.
I wasn't nervous because I've met Hjalmar that did the operation.
Yeah, the surgeon.
And I met Håkan. I got full confidence for my doctors.
Okay.
Oh, no worry.
So that's important. So you felt very confident with the medical and surgical team looking after you, that they were the right people telling you what to do, and you had confidence in them. What about the rest of your family? How did they view this?
We haven't really talked about it, but
Okay, but were
There-
they,
there
was
was-
they as positive? Were they very encouraging of you doing it? Because sometimes I found with some of my patients, some of them are very keen, but the family are less keen.
my family they were positive too. They want me
Okay.
to be better.
Okay.
So it was a positive reaction from my family.
So you went and had this procedure, you know, which was, when did you have the transplant, Michael?
2016,
So 8 years ago. So 2016.
January. First operation was in January
And when was
2016.
the second?
On my birthday,
Oh,
2016. December 7, I was at the hospital, and then December 8, I was operated.
okay. What a birthday present.
What?
So, so you had one in January and one in December of 2016.
Yeah.
And that's 8 years ago, as we were just saying. And how have you been over that 8 years? What's been your response to this transplant?
I can't really say, but I think about how would it been if I didn't have done the operation.
Yeah.
That's what I'm thinking about, because I can't really say how it has done for me, if it's better or I think I've been much worse than I am today.
So if I said 3 things to you, do you think since the operation you've got worse, you've got better, or you've stayed about the same? Which one of those would you think best describes?
I stay the same.
Stayed the same. Okay. So in a condition which is said to get progressively worse, you've actually stabilized for 8 years. And has your medication changed? Have you changed your tablets, or are you still on the same dose of medication that you were 8 years ago?
I'm on the same dose with
Okay.
some changes of the other medicines that I take for. I take about 5 pills on the morning, and then one every third hour.
Okay. So the big thing for you with the operation is you went into it a very positive and a very open mind, and it's really stabilized you for 8 years. And obviously, you know, you're amazingly brave for going forward for this. And obviously it's wonderful that you can share your story with us today. And I thought what might be quite interesting is, you know, there'll be different types of people listening to this webinar. So there'll be scientists, there'll be patients, there'll be caregivers. So what would be your message to the scientists? Because obviously a lot of people work in the lab trying to develop these type of therapies. Would you have a clear message to them?
Keep up the good work.
Keep up the good work.
Because it's nice that somebody likes to work with it, that I'm grateful for.
Okay.
So keep up the good work.
And what about those patients and people living with Parkinson's and caregivers that we have? What would be your message to them?
Thank you to all the caregivers, because it's a good job they're doing. It's nice of them.
Yeah.
And other patients, keep it positive.
Yeah. Yeah.
You can't do anything about it more than just be positive.
Yeah. And
I think.
And presumably, you know, being part of having access to this was very important to you. Did you ever have any doubts when you had sort of been approached by it that this was the right way to go? And did you feel very privileged that you'd been part of a trial? I mean, some people sort of feel slightly frightened by it.
Yeah, I feel definitely privileged, because I had a friend that was in this trial too, and he didn't get the operation.
Yeah.
But I haven't heard from him since.
Okay.
Yeah.
Okay. Well, thank you very much for sharing your story with us, Michael. We've got time for a few questions. So you know, one of the questions we have here was, and I think you've sort of answered this, was, you know, the operation didn't make you better, and there, you know, we discussed before the reasons for that, and Agneta touched upon it, is, you know, the number of cells we could transplant was probably, as it turned out, to be less than the optimal number. But were you disappointed with the outcome of the trial?
No, because I don't know how it would have been without it.
Yeah.
No, I wasn't disappointed.
And then people are quite interested about, you know, when you had the operation back in 2016, what sort of stage were you at with or how far had you come with your journey with Parkinson's? I think, if I've understood it correctly, you know, you'd had it for 10 years.
Yeah.
The drugs were working, but you were fluctuating with the medication. So sort of mid-stage, would that be?
After the first operation, I had doubts, because I woke up with a headache that Metallica had a release party in my head.
Right.
So I had definitely doubts about doing the second, but I had to do it. So there's no talk about it.
And one of the great problems with the fetal transplant trial was that there was great delay in getting enough tissue in order to do the transplant. Did you have many cancelled operations before you had your first and second transplant?
About 15 or 16 cancellations.
Yeah, yeah.
But
So
only twice I was at the hospital and they canceled. So before
right.
that, they called me the day before and said it doesn't work.
Yeah, yeah,
So, but I didn't give up.
yeah. And forums like this where you can discuss your case, do you think this is a good thing for people living with Parkinson's who've had these procedures to go out and talk to the wider community?
Definitely.
Yeah,
I think it's a very good thing.
yeah. Because I think a lot of people don't do this, and you're a fantastic advocate for, I think communicating, what you've experienced with your Parkinson's, and then undertaking these sort of experimental therapies. And are you still followed up in Lund? Do you still see the same team there?
Yeah, every two times a year.
Yeah.
Every 6 months.
Yeah.
I go and do the tests.
Yeah.
So.
And you also had scans, I think, because as Agneta was saying, looking at dopamine in the brain and such like.
Yeah, in London. I went to London a couple of times and did scans.
Yeah. And how did you find that?
I like London. But
it's not a great bit of London where you go and have those scans because it's next to Worldwood Scrubs, which is the prison. But
the problem was for 90 minutes to lay still. 90 minutes don't move.
Yeah.
But that was a problem. But I got Parkinson. Can't stop moving.
Yeah. Yeah. Well, I just want to thank you, Michael, for joining us today. I mean, you're an extraordinary man who tells an extraordinary story, and we're enormously grateful for you sharing that and also putting yourself forward for these experimental therapies and then being completely honest about how they've impacted on you. And I think, for us in the field who've worked on this for many years, it's wonderful to hear your story, and it's wonderful to hear that things have actually stabilized since the time that you've had your transplant, which would not be what you'd expect in people with Parkinson's. From someone who sees a lot of people with this condition, you would expect to get worse. You would expect more medication to be given. So thank you very much for joining us, and thank you very much for sharing your story, and thank you those who've asked us questions.
Thank you. Thank you, Roger.
Thank
Bye-bye.
you. So now it's time for our final talk. And I'm very pleased to have another long-standing colleague and friend, Vivian Tabar, from the Memorial Sloan Kettering Cancer Center. And Vivian is going to talk about clinical trials, future outlook, and emerging challenges. So thank you very much, Vivian, for talking to us today.
Thank you very much, Roger. It's a pleasure to be here. Special thank you to Michael for his terrific testimony. He and patients like him are the inspiration here. So I assume you can see my slides. If not, you'll let me know.
Perfect.
Thank you. These are my disclosures. I'll be talking to you today about a trial for which I was a participant in the design of and the discovery of the science, design, and the implementation. And this trial is supported by Blue Rock Therapeutics. I'm not going to go over the experiments and data leading to the trial in any detail because Agneta covered that very well. Our product is very, very similar to Agneta's group product in that it's an embryonic stem cell that was differentiated into dopaminergic progenitors, underwent quality testing, quality assurance, quality of the cells, function of the cells in the lab. And then in preparation for what we call an IND application, which is a documentation you go to the FDA with, we performed so-called IND enabling studies where first we checked, we've made, as Agneta did, we made a frozen product. So we had numerous small vials containing frozen dopamine progenitors. So the testing happened on the very same lots of cells that will eventually be used in the patient, which is the advantage of this approach. We then conducted efficacy studies. So we put these cells in the brain of a large number of rats and tested if the cells made them better. And then we did other safety tests. We asked if the cells distributed past the area where we injected them in the brain, and we found that they did not. We looked for toxicity on the whole body of the mouse as well as in the brain. And of course, we looked for any tumor formation in the mice. And all the tests were very favorable. There were no tumors, no toxicity related to the cells, and no biodistribution beyond the brain. And this is an example of what things look like. In this picture in the middle panel on top, you see a section through a rat brain. One side has no brown, that's the Parkinsonian side, has no dopamine endings. And then you see a section immediately below it showing surviving human dopamine neurons derived from ES cells that we grafted in that site. You could see the graft survived and it innervated the striatum. And these graphs on the side demonstrate improvement in one particular behavior that Agneta illustrated called the rotation test. You can see that the animals that received the graft improved, while the other animals that did not receive the graft did not improve. So we packaged all this, and a very important next step is how to develop a clinical trial. As a matter of fact, while we were doing the science, when we still were not too sure about the end product and the detail of it, we assembled a team that included not just the scientists who are obviously critical, but also engineers who thought about how to deliver the cells, brain surgeons, neurologists, statisticians, specialists to answer questions about immune suppression, and ethicists to answer questions about the ethics of what we're doing, the cells of origin, the actual building of the trial, to ensure that it's compliant with with rigorous standards of science, but also rigorous standards of ethics. We also thought it was extremely helpful for us to include patient representatives and patients, as well as patient advocates and caregivers for the patients. And so this entire team then thought about first, how do we design this trial in terms of who should receive the graft? Remember, this is a phase 1 trial. That was the first line item, if you will. So phase 1, that's the complying with regulatory guidelines that say before you give this product to a large number of patients, let us put it in a small number of patients to indeed confirm beyond the animal studies that it is safe and tolerable in humans. And so that imposes a different mindset where the primary endpoint that is allowed, meaning the kind of conclusions you can derive from this trial, will be really focused on this endpoint of safety. We, of course, took into consideration all this rich history that Roger and Agneta discussed, meaning the fetal stem cell, from which we learned a lot. And we held ourselves to high standards of scientific rigor and the ethics, as I mentioned. But in addition to the safety primary endpoints, obviously, we built in some so-called efficacy signals. So there were testing also to try and give us an idea if it is likely that the product that we grafted in the patient is actually helping. I'm mentioning this before I show you the data because I want to put it in context. This is quite a complex type of studies where conclusions have to be very measured and discussed accordingly. And that leads us into the importance of setting expectations, both in terms of our communication with regulatory agency, within our own team, but also to the patients, to the scientific and clinical community. And this is one of the challenges, really. I mean, we've been talking about the role of stem cells for a very long time. We're very excited that we're going through this period of actually doing this in the patient. But I feel that a very important step here is to be very clear on expectations. Parkinson's is a very complex disease. Roger explained that we're not reasonably targeting all aspects of Parkinson's. And so keep that in mind as we go through the data. So that goes into this subtitle of recognizing limitations. And at the same time, even though this was a trial on a small number of patients, in our case, 12 people, we wanted to be sure to lighten the burden on the patient. And of course, there's a financial burden to keep in mind, although these early phase trials are covered by the sponsoring companies that we mentioned. So what did our clinical protocol look like? So it was designed to assess safety and tolerability of the procedure. The product was the human ES-derived dopamine neuron progenitors that we grafted in the striatum or the putamen in Parkinson's disease, people with Parkinson's disease. 12 patients, 2 surgical sites, a site here at my institution in New York where 9 people were grafted and 3 people were grafted at the University of Toronto. But the patients were recruited in New York City, at Cornell, NYP, in UC Irvine, and in Toronto. Our trial was designed in 2 doses, and we'll come back to how do we think about dose. A low dose of about 1 million cells and a high dose of 2.7, almost 3 million cells. And we came up with these numbers because we reasoned that we would ideally like to replace the same number that we have in a normal substantia nigra would have. So that's roughly 400,000 cells or so. We calculated based on 300,000. And then we wanted the low dose simply because we did not know if putting in that many dopamine neurons as a target would be the right thing to do. We talked about the concern about dyskinesia happening due to the graft. And the common thinking is that perhaps that was a result of contaminating cells like serotonergic neurons. Some question about dosing also is discussed in the setting of graft-induced dyskinesias. We, in general, excluded commonsensically things like very ill patients, people with serious cardiac or lung disease. But as related to Parkinson's, we excluded patients who had dementia and patients who had significant dyskinesias as reported on the Ames rating scale. What did things look like from the patient's perspective? Some of the patients were recruited from an existing observation study that my colleagues, Drs. Henchcliffe and Sarva had, where patients were enrolled and simply their disease progression recorded, so testing them regularly. And they were aware of the trial coming up. So some patients came from that cohort and others were patients that reached out, and everybody would then be screened for eligibility. There's a longer list of eligibility criteria than I summarized. The patients had an MRI to make sure there are no other concerning issues in the brain and underwent a fluorodopa PET, which is similar to a CAT scan, except you're given a actual 18-fluorodopa in the vein, and we can see where there is uptake in the brain. In Parkinson's disease, we expect to be relatively low uptake compared to normal. Then the patient comes into the hospital, we call that day 0, where they receive the surgery. They're usually discharged home. In this trial, we discharged them home the following day. As Agneta mentioned, our product is called an allograft. It's a human product, but it's coming from a person who is foreign to the individual whom we're grafting. So we also decided to do immune suppression for about a year to protect the graft, particularly around the time of surgery. And the patients then received MRIs at regular intervals in the first year, 3 months, 6 months, 1 year. They also had a PET scan at 1 year, at 18 months, and at 2 years. And of course, they were followed regularly with clinical testing using the various Parkinson's disease rating scores. Again, the primary endpoint was set at 1 year for us to determine if indeed the trial was safe and tolerable. And then we had secondary and exploratory endpoints that are clinical. So how is the patient doing essentially on motor and non-motor outcomes that we are reporting out at 2 years. Today, I'll show you all of these data, missing only the PET images at 2 years, which at the time I made this talk were not yet ready, but should be ready very soon. These are the medicines that we gave for immunosuppression. We started it during the surgery. There was an IV dose of 2 medications during surgery. And then the patient stayed for 1 year on 2 medicines, one called tacrolimus, commonly used in transplant surgery for other organs, and a small dose of steroids. Now, what about the surgery? The surgery is called stereotactic surgery, a procedure that neurosurgeons do commonly. It involves drilling a small hole in the skull and passing a thin cannula into the target, in this case, in orange here, the putamen. Now, we would like to put in these cells in a way that they are distributed reasonably evenly in the putamen. Because they are dopamine progenitors in the late state of being a progenitor, so about to differentiate fully, we don't expect them to migrate. And in fact, we know that they don't migrate far from our animal studies. And therefore, we needed to put the cells in the putamen, which is a relatively large structure in the human, in several areas. So we balanced that need out versus the risk of causing bleeding every time we pass a cannula in the brain, and decided on 3 trajectories. 3 trajectories, 5 trajectories is really relative, but that's what we went with. And along each trajectory, we placed 3 cell deposits. So overall, the patient received 9 cell deposits total in 3 trajectories on one side, and we did the same on the other side of the brain. Notice, as Michael was telling us, he had to wait a whole year almost between surgeries for the right side, left side, depending on availability of cells. But here, having the cells already frozen and available makes the procedure all in one much simpler. I'm showing you a little bit of how this went. In our case, the procedure happened with the patient inside an MRI scanner. The cells were brought into the operating room, loaded up in a cannula, and we traced the trajectory of how we want to send that needle into the brain to optimize safety using software and computer systems that are commonly used in surgery. And this is an example of one patient where we're showing you a little bit of a 3D-like representation of the trajectories and where the cells were placed within the putamen. This is a picture showing the path of the trajectories that we could see in real time at the time of the procedure. So now let's look at the data. So the patients were enrolled in the trial roughly once a month. We had a patient, and so we performed the surgery on all 12 patients. All 12 patients remained enrolled in the trial. In our group, we had an average of about 67 years of age. Last enrollment was, our last surgery was in March 2022. The patients had PD for roughly 9 years at the time we performed the transplant. The Hearn and Yahr staging is basically patients with moderate PD at the time of moderate to advanced PD at the time of surgery. And then these are descriptions of what were their scores on the UPDRS, which is a very commonly used motor rating score for PD. We had two cohorts, a low dose and a high dose, and these clinical features were similar between the 2 doses. As you can see here in a little bit more detail, we happen to have more men than women in the trial. And our first important endpoint is our primary endpoint of safety. So to determine if the trial is safe, we used 3 general categories. Clinical picture, obviously. Also, we reported all side effects, whether we thought they are, as you do in all these trials, whether we thought they're due to surgery or to the cells. And of course, any problems on an MRI or problems during surgery. So I can report that all the patients received all the planned cell doses, tolerated the immune suppression for 1 year. So that was stopped exactly at 1 year. There were no deaths, no bleeding in the brain, no growth of tumor or any other abnormalities on all the MRIs up to 2 years. Another important milestone here is the development of dyskinesias related to the graft. Now, you'll remember that we excluded patients with significant dyskinesia, and that allowed us to have a better sense of whether graft-induced dyskinesias would develop. And as you may recall, that was a concern in the fetal cell transplantation. Within the first year, I believe most of the dyskinesias manifested. So up to 2 years, we do not have any dyskinesia. And most of the side effects were related to, GI upset and mild headache, nausea, related largely to the medicines, the immunosuppression medicine. We had one patient develop COVID, which we did not think was related. This happened during the pandemic, this trial. There was one gastrointestinal hemorrhage that was managed conservatively that we did not think was related. We did have one patient have a seizure, a single seizure the day after surgery, even though their scan was normal. And that is something that is also reported in the DBS literature. That patient received medication for seizures for about 3 months and then stopped, and he has not had another seizure since. This is the actual data, for those of you interested, of the dyskinesia rating scale, demonstrating the absence of development of dyskinesia throughout the 2 years of observation that the patients were on. All the data I will show you is divided between low dose and high dose, low dose in the light blue and high dose in the darker blue. And here are some of the clinical data. First is the Hauser diary. PD patients who are listening and their caregivers know that this is a diary that the patient keeps. They describe the amount of time they spent on, meaning doing well, and off time, doing not as well. You would like to see the on time increase and the off time decrease. Now, I want to be careful interpreting these data. As you can see, I'm showing it with all these long bars, which include the variability. But in general, one can say that there was a trend for less time spent off and more time spent on compared to the baseline. And again, I'm calling them trend. We did not do tests of statistical significance because these are a small number of patients. But overall, you can say definitely the patients remained rather stable, and some of them showed some evidence of going in the right direction. Then this is another very important secondary endpoint, the UPDRS Part 3. And again, this is tested by the clinician on medicine and off medicine. As you can see, there was an improvement or a trend towards improvement in the UPDRS of multiple points, more so in the high dose than in the low dose. This is another part of the UPDRS Part 2, which has motor experiences of daily living. And there again, the trend was towards stability versus improvement, and more obvious in the high dose. I am often asked about Parkinson's disease medications. We did not ask people to change their medications. We left it up to the treating neurologist to respond to medication adjustment. And in general, the medications seem to have remained relatively stable. And this is the PET scan I was describing before. So this is a composite image of all the patients at baseline, at 1 year, and at 18 months. And as you can see, there is a trend for improvement in fluorodopa uptake in the putamen. Normally in a Parkinson patient over a year or two, you would see a continued decrease in dopamine uptake as the neurodegeneration continues. Seeing it to be stable or a little better is a good sign. Again, big caveats here. The PET scan alone is not necessarily an indication of efficacy, but it is an important readout that at least tells us that the graft perhaps survived at the place we placed it. This is an example of the MRI images, all of which were within normal. You will notice these white lines are expected. This is where the cannulas pass through the brain, but this is not a negative sign or a sign of concern. So in summary, we can say that this trial showed was generally safe and well tolerated in all 12 patients, that the surgical delivery was feasible and safe, that there were no dyskinesias related to the graft up to 2 years later, that there's some evidence of graft survival on PET, and that our exploratory clinical scores may suggest improvement. Importantly, the clinical trend and the DOPA uptake continued to be improved or stable after stopping the immune suppression at 1 year. And of course, we should interpret this with caution in view of the small number of patients. I will not have time to go through challenges and opportunities, but I will list them. We talked about autologous versus allogeneic sources. We are still thinking about the optimal dose, knowing that this is a dynamic live product that may change over time. Should we be placing specific midbrain neuronal subtypes? Would those be more efficacious than current protocols? We talked about the potential impact of the microenvironment on the grafted cells in the long term. Often we ask ourselves whether we should graft in the nigra as opposed to the striatum. And there's lots of pros and cons to this approach, and more science is needed to be confident that placing the grafts in the nigra is a good idea in terms of axonal finding. Neurophysiology tells us that it would be a good idea if it were feasible. We need better imaging strategies to assess graft function. What about the role of wearable devices in monitoring the impact of the graft? We need better surgical devices and cell preps, and we are already talking about how best to design definitive clinical trials. At the stage for the Blue Rock trial, there is a phase 2 planning to come on board later in 2025. And Agneta already discussed all the ongoing clinical studies or other trials in the US and elsewhere. And of course, this was a huge team effort, and we thank the team and our funders.
Thank you, Vivian. Absolutely superlative talk. We've just got time for a couple of questions. If I can just ask a couple of questions which have appeared, Vivian. I don't know if you can reappear with your camera. So there were a couple of questions about the surgery. So
Yes.
one was about the duration of surgery, and related to that is obviously the number of tracks and deposits. So I don't know if you could just say in your trial how many tracks you used to put the cells in, how many deposits, and how long the surgery took.
Yes, we did 3 tracks per hemisphere, 3 cell deposits per track, so 9 total on each side, a total of 18 cell deposits. The duration of surgery, frankly, varied. The first couple of patients was very long. We were super nervous, cautious, and then once we got our workflow going, we were down to roughly 3 hours for the surgeries that happened in the MRI scan. I believe my colleague in Canada did it in a little bit shorter with a more traditional frame-based stereotaxy.
Okay, and one of the questions was about, this is a cryopreserved product.
Correct.
So when you get it out of the freezer, presumably you have to then prepare the cells for surgery. So how involved is that, and how long does that take? And is that something which might be a limiting factor going forward with this approach?
So we had a GMP facility where these cells were frozen on site. The team would come in early morning and thaw the cells, wash them, and prepare them, meaning suspend them, in the right concentration. Then we had to perform an endotoxin test on them, which takes about half an hour. So altogether, this part, again, in the beginning took longer, eventually came down to a much shorter process, but still I would count at least an hour for the cell prep, if not more. I don't think it was limiting at all. We had access to more vials if we needed them, if there's something that happened that we weren't happy with, so.
Okay, and the final question, which is the question I always get asked and has appeared in the question and answer, is when do you think this would become a standard of care? So assuming these trials work, when do you think a patient, someone with Parkinson's, can go and say, I don't want DBS, I don't want your drugs, I want that cell therapy? When do you think that's likely to occur if the trials prove successful?
Yes, there's an if and a when to this question. You know, we are on about to initiate larger trials. So the next phase 2-3 will have 50 or 60 or 70 more patients, which is going to be super exciting. We'll have a lot more information. I don't think it'll happen. If I want to answer the question straight on, I don't think it'll happen before about 5 years if it happens, because we need to monitor the patients long enough. But there's huge excitement and multiple teams doing this. So it will happen in the near future, hopefully.
A politician's answer. Thank you so much.
I just said
thank you. Thank you. I would just like to, just conclude our program now. So I'd like to thank Vivian. I'd like to thank Agneta. I'd particularly like to thank Michael for their insightful presentations, engaging discussion, as well as our webinar co-sponsor, Blue Rock Therapeutics. I would love very much to thank our audience. You've been absolutely spectacular in the questions. So I'm sorry we haven't answered everything, but we've been trying to answer all the many questions that have come forward. So thank you for attending. Today's program will be available soon on demand on the ISSR and WPC website. Thank you all for joining us. Have a great day. And here's a little bit of information which you can digest before you disappear. So thank you very much.